Thank you so much, Flemingsberg Science, for inviting me to give this talk, and thank you all to be here. I'm not sure if you came for the talk or the breakfast, but now you're stuck with me. So Truly Labs, we provide preclinical services within the drug development world. But maybe before I can tell you a little bit about me, my name is Carla. I'm Portuguese, did a master's in pharmaceutical science, got the amazing opportunity to come to Sweden to do a PhD in Uppsala in renal physiology. Then life dictated that I travel down south to Skåne, where Truly Labs found me, and I started working there together with 25 amazing colleagues. We have roughly 300 years of experience in drug development. I think I pressed this controller. We've been active for 15 years now. We've recently moved to this beautiful building here. It's where we have our new in vitro facility and offices. And within this time, we've delivered more than 5,000 studies to more than 230 different customers. A few of them in Stockholm, a few of them within this area as well. We like to think that we are a small to medium fish in a very big sea, and together with other companies, other service providers that we can collaborate with, we can actually meet up the demands and match up to what can be considered the bigger fishes or the big pharma. United we are strong is what we believe in. So enough about us. Just for reference, I think it's always important to talk about our mission and vision, and you have it here. Our vision is to create opportunities for the next generation drug development to improve health for patients, and our mission to improve truly transnational preclinical service characterized by the highest ethical and scientific standards. Our core values are trust, teamwork, and truly engaged. And with this, we move on to what do we offer. So we're a full-service strategic partner in drug development. We offer preclinical pharmacology. We work also in precision medicine and give strategic advice. Here you have a very big overview of our technologies and service. We do cell-based assays. We have ligand binding immunoassays as well, molecular assays, histology and imaging, microbiology, and we have also quite a good inhalation service packages, and we do in vivo. When it regards to tissue and cell preparation, we do have our own BSL unit 2 as well. We have primary cell lab and different cell lines that we work with. Applications for this can be various, from cell cytotoxicity and viability, protein or antibody binding, internalization, biomarker screening, etc. We do a lot of flow cytometry, as, for example, readouts for the in vivo studies that we perform. A lot of phenotyping, functional assays, cell proliferation, viability, receptor analysis, receptor density, etc. And we do work with different sample types, from cells to lavage fluids, tissues, and of course blood. When it comes to bioanalysis, we are a certified service provider for Olink, so in case you're interested about that. We also run quite a lot of multiplex immunoassays and your normal ELISAs. We also we can also create or make the assays that you need or develop the assays that you need in case there's no commercial kits available. When it comes to histology, we have a full service pack, beautiful images. We have the cryosectioning and the paraffin embedding and sectioning, the normal conventional histological stainings like H&E. We do immunohistochemistry and immunofluorescence, proximity ligand assays, and we also work with the RNA scope and in situ hybridization, as you can see this beautiful picture. We work with different types of tissues as well. We also quantify some of the stainings. So we have quantification here of lung metastasis as an example of our workflow in terms of percentage of stained area calculated over the area of interest. So this is something you can get. When it comes to microbiology and infection, we work both in vitro and in vivo. We also have a BSL-2 in vivo laboratory. We work with different bacterial species. We do in vitro studies, CFU, local and systemic analysis of cytokines, etc. And we also have some in vivo models in rats and pigs. Pigs is in a collaboration. We have imaging as well. We have the Pearl for using with labeled bacteria, and we can also use the IVIS as well in collaboration. We are strong in immuno-oncology, where we have different tumor models, and we can test different drugs as well. We have syngeneic tumor models and CDX, and we can also do co-cultures. Here you have examples of a treatment of a syngeneic murine triple-negative breast cancer. I believe this was with cisplatin, where you see a reduction in the tumor growth. You have visualization with a Pearl of a tumor growing, and then some flow cytometry analysis of tumor cells. We do a lot of other things that I'm not showing here, but here's a few examples of acute inflammatory models that we've worked on. We have, for example, IBD model, inflammatory bowel disease. We have a carrageenan poedema, which is quite an interesting one to test the effect of anti-inflammatories in reduction of this swollen little paw. And we also have the MSU-induced peritonitis. We love PK. We think that pharmacokinetics and pharmacodynamics are very important in drug development. Some people might not realize that, and we always try to tell our customers that this is important. We run quite a few PK studies. Unfortunately, I don't think we run a lot of PD studies. That's for different reasons. It's a little bit more tricky to do a proper PK-PD study. But it's important if you can do them. And as I mentioned, inhalation is also one of our biggest strong points. We are part of MVIC, which is the Medican Valley Inhalation Consortium. We have quite a lot of animal models for inhalation where we test PK, PD, lung toxicity, lung disposition and distribution. We administer compounds via different routes, intranasal, intratracheal, nose only, or even whole body. And we have different readouts for low and systemic biomarkers, immune profile of the lung tissue, lung pathology, etc. And we can do this in mouse, rats, and sometimes a little bit larger animals as well. And I also wanted to pinpoint that it's not everything in vivo. We also try to reduce the number of studies and actually point more towards in vitro analysis. And we do offer, here an example, the air-liquid interface cultures, in which transepithelial electrical resistance can be measured to visualize the integrity of the membrane. And you can use this to test toxicity of inhaled products. And with that, I leave you with our website. It's a new one, launched very recently. Please check it out. You have a lot of different contact informations there depending on the services you're interested in, and also my email. And thank you.